Good evening, everyone. Myself, Dr. Aanchal, and as we have started the series of renal physiology, so today we are having class fourth, and the topic have that is counter current mechanism. So before proceeding, let's discuss some core features of Oracle. So in Oracle, we provide live classes in which there will be direct interaction of the student with the faculty. And live classes is basically for the elevate learning of the student, and here knowledge meets interaction. Then recorded classes are also provided by Oracle in which the student have access of all the subjects anywhere, anytime through the recording sessions. Then also Oracle provide ultimative comprehensive book with CVME based question answers, MCQs, and full zone for fun zone for self assessment. Then in Oracle, we also provide MCQs, and MCQs are from beginner level to expert level, contain standard multiple choice questions. Then student can also book a slot for personalized counseling session in which there will be direct interaction of the student with the counselor. Then Oracle will provide some subscription plans. So basically, we have three plans starting from basic to standard and then premium. So basic plan is of 3 months, which cost around 15,000. It includes pre-recorded classes, live classes, and quick revision examination module. Then in standard plan, we it is of 6 months, cost around 30,000. It includes pre-recorded classes, live classes, doubt clarification, quick revision examination module. Then final plan we have that is of premium plan, which is of 12 months and cost around 50,000, and it includes all the services that is provided by Oracle, in which we have pre-recorded classes, live class, live doubt clarification, one-to-one personalized guidance notes, and quick revision examination module. So I would recommend all the student to take premium plans because it includes all the features that are provided by all the services that are provided by Oracle. And also for 20% instant discount, you can use my code that is Aanchal20. So as we have started the renal physiology, and today we are having class 4 that is of counter current mechanism, and further classes we are having on 17 March acid base balance, then maturation on 18, and then dialysis and diuretics on 20th March, and renal failure and RFT on 27 March. Okay, then here we have the clinical case scenario that we're going to discuss in the end of the session. So basically, when we talk about glomerular filtrate, so glomerular filtrate is approximately 180 liter per day, right? Glomerular filtrate is 180 liter that is formed with large quantity of water. And if this much of water is excreted in urine, so obviously body will be in a serious threat, right? So that's why of this 180 liter, 99% get reabsorbed and only 1% is there for the formation of urine, right? So the concentration of urine is also very essential here. Now the osmolarity of this glomerular filtrate is same that of plasma, and it is 300 milliosmo per liter, right? But normally urine is concentrated and its osmolarity can be ranged between, the osmolarity of urine can be ranged between 50 to 1200. So basically this osmolarity will totally depend upon the water content which is present inside the body. If there is much water content is present, then the urine will be more diluted and its osmolarity will be around 50, 100. And if the body is facing the condition like dehydration, so in such condition, the urine output will be low. Secondly, and the urine will be more concentrated and it can goes up to 1200 milliosmo per liter. So for this mechanism, for this dilution and this concentrated mechanism, basically we study the very important mechanism that is countercurrent mechanism that occurs between loop Henle and vasa recta that we're going to discuss today, right? So when there is excess water in the body and body fluids, osmolarity is reduced. The kidney can excrete urine with an osmolarity as low as 50, a concentration that is about 1/6 of the osmolarity of the normal extracellular fluid that we have discussed. It is about 300. And when there is deficient of water, means the body is in condition of dehydration. So at that time, the concentration of urine can be about 1200, or maximum it can be 1400, right? So it will totally depend upon the water content that the individual is taking, that whether the urine will be more diluted or it will be more concentrated, right? Now, next we have that osmolarity of water basically depends upon two factors. First, we have that already we have discussed, the water content that is present inside the body. And the second, we have the role of ADH, means antidiuretic hormone, or that is also known as vasopressin, right? So, mechanism of urine formation is same for dilute urine and concentrated urine till the fluid reaches the DCT, or collecting tubule. Okay, so dilution or concentration of urine depend upon the water content and ADH also. Now the control secretion of ADH, which regulate the permeability of DCT and collecting duct to water. A high osmolarity of the renal interstitial fluid, which provide the osmotic gradient necessary for water reabsorption to occurs in the presence of high level of ADH. Now basically how it happened, what is the mechanism, that we're going to learn. Now, let's discuss the different type of nephron that is present inside the kidney. So as we know, there are two type of nephron there. First, we have the cortical, and the next we have the juxtamedullary. So when we look at the diagram, so if the kidney has two parts, the outer is cortex and the inner is medulla, right? So when we talk about this cortical nephron, so basically the glomerulus part of the cortical nephron lies in the glomerulus part is lies in the upper region of this cortex part, and its loop of Henle lies in outer layer of medulla, right? And if we talk about the juxtamedullary nephron, so we know that glomerulus is near the junction of cortex and medulla, as we can see in the picture, and its loop of Henle is present in its own medullary part. And second thing, the loop of Henle of cortex is smaller than the loop of Henle of juxtamedullary nephron. So the LOH of juxtamedullary nephron is always larger, or we can call longer in juxtamedullary nephron. And that's why these type of nephron are mainly involved in the countercurrent mechanism. So let's look for the difference part. So as we have discussed, as we have seen in the picture also, in cortical nephron, the glomerulus is present in the upper region of cortex that we have seen. The glomerulus in juxtamedullary nephron is present near the junction of cortex and medulla. Yes. Then talking about the number, so they are approximately 85%, and juxtamedullary nephron are 15%. Then the glomerular size is smaller in cortical nephron, and it is larger in juxtamedullary. And as we have seen, the loop of Henle small up to layer of medulla, and the loop of Henle large deeper into the medulla, as we have seen. Then in cortical nephron, we have ascending limb, the thick segment, and in the juxtamedullary, we have a thin segment of ascending limb. Then talking about the efferent arterioles, large diameter and forms the peritubular capillary network, right? And talking about the juxtamedullary nephron, in efferent arteriole, basically it has small diameter, and here it forms the vasa recta, which is also responsible for current mechanism, and here it acts as a exchanger, right? Now the rate of filtration is quite slow in cortical nephron, and in juxtamedullary we have a high filtration rate. Okay, now talking about the very important difference, that is difference in the function. So in cortical nephron, the major function are the excretion of waste and products in urine, whereas in juxtamedullary nephrons are mainly responsible for the concentration of urine by countercurrent mechanism that we're going to learn today. So I hope this difference between the cortical nephron and juxtamedullary nephron is very well clear to all the students. Now, talking about the countercurrent system, so it is a system in which the inflow and outflow runs in close proximity and parallel to, but in opposite direction. Now, why they are calling it close proximity and parallel and in opposite direction, that we're going to learn with the help of the diagram. So we have some components of the countercurrent mechanism. So for the component part, we have countercurrent multiplier, which includes descending and ascending limb of loop of Henle, DCT part, collecting duct, and corticomedullary gradient that we're going to learn with the help of the diagram, that where these components are present. Then also the countercurrent exchanger, for the exchanger we have vasa recta, and why this is known as exchanger, that also we're going to learn. Then principal solutes, we have sodium and urea, and the hormone, a very important hormone, the role of ADH is there. Now, now let's talk about the different components that already we have discussed, right? So as we know, a countercurrent system is a system of U-shaped tubule, okay, in which the flow of fluid is in opposite direction in the U-shaped tubule that we can see, right? So for the countercurrent multiplier, we have, for the multiplier, we have loop of Henle, and for the exchanger, countercurrent exchanger, we have vasa recta. And vasa recta is present in juxtamedullary nephron. Okay. Now, this loop of Henle function as a countercurrent multiplier, and it is responsible for development of hyperosmolarity of this medullary interstitium. So here we have medullary interstitium. This is, here we have medullary interstitium. Okay. Now we're going to discuss the role of loop of Henle in development of medullary gradient. Okay. So first of all, we should know that what is medullary gradient. So basically, this is a type of gradual increase in the osmolarity of the medullary interstitial fluid. So whenever there will be increase in the medullary interstitial fluid, so that is known as medullary gradient. And also it plays a very important and significant role in the concentration of urine. So loop of Henle of juxtamedullary nephron plays a very important role in the countercurrent mechanism, because the loop of Henle is long, that we have discussed, and extend up to the deeper part of the medulla. Right now, what are the events that occurs during the development of medullary gradient by countercurrent multiplier? Right now, we're talking about the role of loop of Henle only, multiplier. Okay. In next slide, we're going to discuss about the exchanger part, means the role of vasa recta. So the hypoosmolarity here, the hypoosmolarity of this medullary interstitial fluid is due to active reabsorption, that we have discussed, the how the active process of active reabsorption occurs, that we have discussed in the urine formation of sodium chloride, active reabsorption of sodium chloride, and also other solutes from this ascending part. Okay, as we already know that the descending part is permeable to water and impermeable to salt, whereas this ascending part is permeable to salts and impermeable to water, right? So there will be active reabsorption of sodium chloride and other solutes from ascending limb of loop of Henle into the medullary interstitium. Here the reabsorption will occur, and these solutes accumulate in the medullary interstitium and increases the osmolarity here. Okay, as we can see, there is active reabsorption of NaCl is occurring, right, from ascending segment. Now, due to the concentration gradient, the sodium and chloride ions diffuse from medullary interstitium into the descending limb of loop of Henle and reach the ascending limb again through the hairpin vein. Now, the sodium and chloride are repeatedly circulating between ascending and descending limb of loop of Henle through the medullary interstitial fluid, leaving a small portion to be excreted in urine, right? Apart from this, as we know, that there is regular addition of more and more new sodium chloride ion into the descending limb, okay? Descending limb by constant filtration, because there is constant filtration is also occur. So no more and more sodium chloride also adding, right? So the reabsorption of sodium chloride from ascending limb and addition of new sodium chloride ion from the filtrate increases or multiply the osmolarity of this medullary interstitium and medullary gradient. So that's why this is known as countercurrent multiplier in this way. Okay. Now, okay, we have a question. The countercurrent mechanism operates in nephron. Now every student knows this answer, that it operates in ascending and descending limb of loop of Henle, and ascending limb of loop of Henle, descending limb of loop of Henle, or between LOH and vasa recta. Yes, the answer is between loop of Henle and vasa recta. Okay, so here we are having just give me a minute. Okay, so here we are having a picture through which we're going to discuss the mechanism of this countercurrent mechanism. And for better understanding, I would like to draw here the Bowman's capsule and afferent arteriole, efferent, and forming the glomerulus. Okay. So one by one, we're going to discuss the role of all these components. Okay. So first, we're going to start from this Bowman's capsule. So glomerular filtrate collecting at the Bowman's capsule is isotonic to plasma, and we know that the osmolarity of the this plasma is 300, right? Now this is isotonic to plasma because it contains all the substances except plasma protein. In the last class also we discussed why plasma protein is not present here, because the pore size of this capillaries is very small, and the size of the proteins are large, so that's why they cannot be filtered. So here in the Bowman's capsule, it contains all the substances except plasma proteins. So the osmolarity here of the filtrate will remain similar to plasma, and that is about 300, right? So this is about the Bowman's capsule. Next part we have the PCT part. Okay. So in the last class also, in urine formation, we have discussed that when the filtrate flows through this PCT part, there is active reabsorption of salts, like sodium and chloride. And along with salt, there will be reabsorption of water also occur by some passive processes. So the osmolarity of fluid that is present inside the PCT will also remain similar to plasma, that is about 300, because in this case also both salt and water both are getting reabsorbed. So that's why the osmolarity here also remains same, and that is about 300, right? Now, next we have this part, the descending part and the ascending part. Now, before proceeding, we should learn that thin ascending limb is permeable to water and impermeable to urea. Okay, so this segment, this thin descending segment is always permeable, means there will be more and more absorption of H2O will occur, but no reabsorption of salt will occur. Okay, so whenever the fluid passes through this descending segment, water is reabsorbed from this tubule part into this medullary interstitium. Here we have medullary interstitium. Okay. So the water is reabsorbed from tubule into the medullary interstitium by means of osmosis that already we have discussed. Now it is done to increase the osmolarity in the medullary interstitium. Now the osmolarity of the fluid inside the segment will increase, right? Now, because as we know, there will be, here we have the osmolarity that is about 300. Now, if there will be more and more reabsorption of water will occur, but salt absorption will not occur. So obviously in this segment, in descending segment, there will be accumulation of salt will occur. So from here, the osmolarity of the fluid will going to increase and it will reach up to 1200. Okay, so that's why this descending segment is known as the concentrated segment also. Okay, so this segment is also known as the concentrated segment because in this segment, in this part of the loop of Henle, there will be more and more, because in this part there will be osmolarity of the filtrate will increase up to 1200. Okay, so this is all about the the role of this descending segment of loop of Henle. Now let's talk about this part, ascending segment. Okay, so as we have talked that this part means this descending part is permeable to water but impermeable to salt. Now exact opposing thing will occur in this ascending segment. This is permeable to salt and impermeable to water. Okay, so this is permeable to salt and impermeable to water. So here till this part, the osmolarity reaches up to 1200. Now, because there will be reabsorption of salt will occur and water is not going to absorb. So again, the urine, the filtrate will become more diluted. Okay, so the osmolarity is going to decrease from this part and it is going to reach up to 300 and in DCT we will have up to 100. Okay, so when the thin ascending, okay, one more thing, that in ascending limb we have two segments. We have thin ascending segment and we have thick ascending segment, as we can see. So when the thin ascending segment of the loop of Henle ascends upward and the medullary region, the osmolarity decreases gradually, as we have discussed. Due to concentration gradient, the sodium chloride diffuse out of tubular fluid and the osmolarity decreases up to 800, then 600, 400, right? And talking about the thick segments, we know that in thick segment, we have another channel that is known as sodium potassium 2 chloride. So by this channel, there will be reabsorption of the salts with the help of these segments and the osmolarity will decreases inside the ascending limb, right? Now let's talk about the role of these DCT and the collecting review. So, as we know, while entering the DCT, the osmolarity of the fluid is 100, as we have talked, right? And it is also isotonic to plasma. Now, both this DCT part and collecting tube both are impermeable, okay, impermeable to water. But in the presence of ADH, both will become permeable to water. And why this occur, that we will also discuss in the mechanism. So, in the presence of ADH, both DCT and collecting tubule become permeable to water and finally resulting in more and more reabsorption of water and the final concentration of urine. So here we have learned that as the ascending part extend upward, the osmolarity will decrease, but in the presence of ADH in DCT and both in collecting tubule, there will be active reabsorption of water. So again, the osmolarity will increase. Okay. So starting from here, it starts increasing and it can reach up to 1200. And here it is forming the urine. Okay. Now it is found that in collecting duct, P cells are responsible for ADH-induced water reabsorption. Okay. So in collecting tubule, there are type of cells that are known as P cells or principal cells are present that are responsible for the reabsorption of water, and reabsorption of large quantity of water increases the osmolarity of final gradually, and when urine leaves the collecting tubule, the osmolarity can reach up to 1200 that we have discussed. So thus the urine become hypertonic to plasma. Okay, so in this way. So I hope this mechanism is very well clear to all the students. Again, we're going to discuss it through a quick recap. So as we have discussed, through the afferent arteriole, the blood is going to enter in this glomerular region. And as we have discussed in the last class also, that the plasma protein cannot be filtered. So the filtered, whatever filtered reaches to the Bowman's capsule, the osmolarity will remain similar to the plasma, that is approximately 300. Talking about the PCT part, so in PCT part also, both salt and water get reabsorbed. So that's why again the osmolarity in PCT part is going to remain 300. Now the filtrate will go into the descending segment of the loop of Henle, and we know that this descending segment is permeable to water and impermeable to salts. So there will be reabsorption of water occur, and as the water get reabsorbed, there will be accumulation of salts will occur inside the descending segment. So the osmolarity of the filtrate will increase and reaches up to 1200. Then next we have the ascending limb, in which we have thin segment and thick segment. And in thin segment, there will be a reabsorption of water will not occur because it is impermeable to water and permeable to salt. So again, there will be dilution of filtrate will occur. So because there will be reabsorption of salts will occur from this segment, and the osmolarity will reach up to 100. Now talking about the DCT and collecting tubule, as we know, both are impermeable to water, but under the influence of the hormone, that is ADH, both DCT and collecting tubule become permeable to water, and there will be continuous reabsorption of H2O from both DCT and collecting tubule. So again, the osmolarity of the filtrate will become 1200, and this urine become hypertonic to the plasma. Okay, now let's talk about this part, that is vasa recta. Now, in the arrow, I would like to tell all the students that here black arrow represent the diffusion of NaCl, salts, and this blue arrow indicates the movement of this water. Okay. Okay, fine. So now here the vasa recta functions as countercurrent exchanger. It is responsible for the maintenance of the medullary gradient, which is developed by countercurrent mechanism that we have already discussed. Okay. Now, vasa recta act like countercurrent exchanger. Why it is known as countercurrent exchanger, that we're going to discuss, because there will be continuous exchange of the salts and the water, right? So basically, it is U-shaped tubule with a descending limb and a ascending limb. Okay. And here we have this hairband like structure. Okay. It descending limb runs along the ascending limb of loop of Henle, and its ascending limb runs along with descending limb of loop of Henle, that already we have discussed, that basically this is present like here. Okay. Now we're going to discuss the events that occurs in this vasa recta. Okay. So basically, first, a very important thing, that the blood flow inside the vasa recta is very slow. Okay. And why it's slow, that also we're going to discuss. So there will be, from this descending limb, there will be continuous intake of salt inside. Okay, salts will move continuously. And the water from here move continuously. So that's why, as we can see, the osmolarity will also increases. Okay, from talking about the descending part, so descending limb of this vasa recta, the osmolarity here we have 300, because there is continuous diffusion inside this vasa recta. So that's why the osmolarity is increasing, and the water is moving continuously out of this descending part. So that's why there will excessive accumulation of salts, and osmolarity will reaches up to 1200. Okay. Now from here, as we can see, there is continuous diffusion inside the vasa recta of water, and salt is getting out of it. So that's why the osmolarity will again decreases, and that become up to 300. So that's why there is continuous exchange of salt and water is going on between ascending and descending part of this vasa recta. So that's why it is known as countercurrent exchange. Now again, I'm going to repeat the events that is going on this vasa recta. So as we have seen, the sodium chloride reabsorbed from this ascending limb of a loop of basically this vasa recta, and it enters the descending limb of vasa recta, and simultaneously water diffuses from descending limb of vasa recta into medullary interstitium, that we have seen. Also, we have discussed that the blood flow is very slow this vasa recta. So the large quantity, why this blood flow is very slow? So the large quantity of sodium chloride is get accumulated into this part, into this descending segment of vasa recta. Okay, and flows slowly towards the ascending limb. By the time blood reaches the ascending limb of vasa recta, and the concentration of sodium chloride increases very much, and this causes diffusion of sodium chloride into the medullary interstitium. As we can see, there is active reabsorption of salt back into the medullary interstitium from this part, right? Simultaneously, water from medullary interstitium also enters inside the this descending part, right? This blue arrow indicates it, and the cycle is repeated, right? So basically, why this vasa recta is very important? So, because so that the amount of salt that is present inside the kidney should remain in this medullary interstitium and get exchanged continuously, and whatever the amount of water is present, that is flows with the blood that is present inside the vasa recta into the body, so that the body will remain hydrated. Okay, that is why this mechanism is important. The amount of salt that is present will remain inside the medullary interstitium, and whatever the high and huge amount of water is present will goes with this blood into the body to remain the body hydrated. Okay. Please allow me to share this slide. So we were discussing the role of EDH. So as we can see in the picture that in the presence of EDH, the both DCT and collecting duct become permeable to water, and in absence, it's become impermeable, right? So here it is producing large volume of dilute urine, and here the small volume of concentrated urine is here. So we're going to discuss with the help of the mechanism. So basically, there is a high osmolarity of the renal medullary interstitial fluid, which provide the osmotic gradient for the reabsorption of water. Whether the water actually leaves the collecting duct by osmosis determine the hormone ADH, and the osmoreceptor in the hypothalamus detect the low level of water of high osmolarity, so that the hypothalamus sends an impulse to pituitary gland. As we know, the ADH is secreted by posterior part of the pituitary gland. So the signals are sent to pituitary gland, which releases ADH into the bloodstream. Now ADH makes the wall of the collecting duct more permeable to water. So therefore, when ADH is present, more water is reabsorbed and less is excreted. So we're going to discuss with the help of this diagram. So the final concentration of urine is achieved by the action of ADH. And normally, as we have discussed, both the DCT and collecting duct are not permeable to water. But with the presence of ADH, makes them more permeable and resulting in water reabsorption. And the large quantity of water is removed from the fluid while passing through the DCT and collecting tubule. So the urine become hypertonic with the osmolarity of 1200. And now we're going to discuss that what is the mechanism here, right? So basically, antidiuretic hormone increases water reabsorption in DCT and collecting duct by stimulating the water channels that are known as aquaporin 2. Okay, now what is aquaporin? Aquaporin are the membrane-bound protein which act as a water channel. Okay, and there are about 5 aquaporins are present inside the humans, and aquaporin 2 here forms the water channel in renal tubule. Okay, so basically what happened, this ADH is combines with V2 receptors in the tubular epithelium. Okay, so here we have vasopressin receptors that are known as V2 receptors in the tubular epithelium and activates the adenyl cyclase, so that this cyclase AMP can be formed, which act as a second messenger signal. And here, this cyclic AMP activates these aquaporin so that there will be increase in the water reabsorption, right? So here, as we can see, vasopressin bind to membrane receptors, as we have seen in this diagram, and they activate cyclic AMP, second messenger system, and cells insert aquaporin water pores into apical membrane here, and water is reabsorbed by osmosis into the bloodstream, right? As we can see from here. Okay, so this is how the mechanism of vasopressin occur here. Okay, so this is all about the mechanism that we have discussed, and in the last we have the summary. So the main function of countercurrent system is to concentrate urine, and juxtamedullary nephron plays a key role. And countercurrent mechanism has two components, that is a countercurrent multiplier and exchanger. Countercurrent multiplier generates a gradient in which turn is maintained by countercurrent exchange system. Countercurrent exchanger is also present in skin, testes, and intestine. Sodium and urea are the main solutes, and sodium potassium transporter in ascending are the major pump. Okay, then we have the MCQ, and this for this MCQ all the students will get 30 seconds. Allow me to share the screen. Yes, the answer is reabsorption of sodium in thick ascending limb. Now we have a clinical case scenario also. So the case is that a male present with emergency department with this complaint of excessive thirst, frequent urination, and weakness. On examination, the patient appears dehydrated with dry mucous membrane. Laboratory investigation reveals hyponatremia, hyperosmolarity, elevated blood urea, nitrogen. Further workup indicates a urine osmolarity significantly lower than expected. Which of the following is most likely underlying the mechanism contributing the patient condition? And I know all the student knows the answer. First, we have impaired RAS system, then dysfunction of JJ apparatus, decreased GFR, or disruption of the countercurrent mechanism. Again, you have 30 seconds to solve this. Yes, the answer is disruption of the countercurrent mechanism. Okay, so I hope all the students find the class more helpful. So this is all about the countercurrent mechanism. Thank you.